Hello, everyone. Thank you for joining us. I'm glad you could be here. Feel free to say hi in the chat. Pradeep, hi. Krishna, hello. If you happen to be new, as always, I like to say, let us know where you're joining us from. Today we're going to be looking at some string problems. So one of the things that we've mentioned recently is we're kind of at this beginning stage of, we had some very introductory problems. And now we've got maybe more like advanced beginner problems. And what we're going to see, I think, at the end of today, and then as we go towards our next stream, is a collection of problems that really require us to understand some algorithms at a kind of greater depth, and that require us to build something that requires optimization. So that's kind of a preview of where we're going to get to at the end of today's stream. But the start is going to be, clearing my throat, the start is going to be a lot of problems that are kind of just preparing us for these more advanced questions and just continuing to lay the foundation in algorithms for introductory string analysis. So, Hank, welcome. Thanks for coming. Anna, thanks for coming. The model for these is I always solve these problems and I just kind of walk through them. If you have any questions, let me know. Normally what we do is I will discuss any questions that you happen to have in between. Otherwise, it's just an opportunity for me to just go through these problems steadily, solving one question at a time. And we're going to solve 6 questions today. Well, we'll see if that's true. But I hope you enjoy these streams. We run them on Thursdays at 11 a.m. Eastern. Check out links in our description. We have a lot of other helpful links, but in particular, if you want to follow along, we have starter code and completed code folders that are linked in the description of this video. So check those out if you want to code along with us. Our partner project is, of course, Programming for Lovers, which you can find at programmingforlovers.com. We're currently streaming a lot of code alongs there with learning about scientific algorithms and then applying those to some real data. So we just finished a section on evolutionary trees. We've got another one coming up on parallel programming and distributed programming and applying that to building a chemical simulator. So we're still kind of working on that one a little bit, and it's really exciting because I think I'm the only intro programming course in the world that teaches parallel and distributed programming, at least at any depth. So that's what we'll be doing on those, and those streams happen Monday nights at 7:30 p.m. Eastern. So never too late to join, and you can always join our class at programmingforlovers.com. As always, please make sure that you like and subscribe. It helps keep us visible. And if you want to know about all the cool stuff that we're doing, both from Carnegie Mellon as well as just projects that I'm building out, for example, our Great Ideas in Comp Bio course, which is going to be coming soon. The way to stay connected on all that is to make sure that you're in our Discord and that you join our mailing list. So we have links to that in the description as well. And otherwise, I think we're ready to get started. So thank you all for joining us. Iris, how you doing? Julia, good to see you. And the first problem I'm going to look at today is TRAN. All right. It's a nice warm-up problem, too. Problem TRAN asks us to look at 2 different types of mutations. So it gives us a little bit of biological background here. But the idea is that the DNA nucleotides themselves separate out into purines and pyrimidines. And I will say I always fail to remember at times which pairs of nucleotides are called which. But the idea is that A's and G's are going to have more similar chemical structure to each other than they're going to have to the other nucleotides. And so A's and G's are going to be more chemically similar. C's and T's are going to be more chemically similar. So from this perspective, these transitions between one of the AG nucleotides and a CT nucleotide is going to be less common as a mutation. And so those less common mutations are called transversions. If you're just staying within the same category, it's going to be called a transition. And so what we're doing here is we're taking steps little steps beyond what we've already done. And one of the questions that we've already encountered is trying to determine what is the distance between two strings. So how is it if you give me two strings, I can compare those two strings? And we've done relatively straightforward things. We're going to eventually move towards alignment and see some amazing algorithms for aligning strings. But for now, we have looked at something like Hamming distance. And Hamming distance simply says line up two strings of equal length, count up the number of mismatches, and say how many mismatches are there between these two strings. It's a great first effort. It's flawed because it can't handle strings of differing lengths. It also can't handle the fact that maybe we're going to be deleting or inserting symbols into each of these strings to make them rearrange from one string to another. So we're trying to move more biologically accurate. And for the meantime, we'll say, well, at least what we can do is we're going to take the Hamming distance, we're going to improve it a little bit just by saying, how many transitions do you see? How many transversions do you see? And count them up. And what this problem says is, can you identify the difference between the two? And then we're asking for one number, which is the ratio between the number of transitions that you see and the number of transversions that you see. So in this particular case, when you take that ratio, the transitions are more common, so you're going to get 1.214 for this sample output. If we want to implement this function to solve this problem, then the first thing that we might do is to just establish, well, we probably need to make sure that we're keeping track of the number of transitions and the number of transversions between these two strings. And one of the things that's nice about Rosalind is we're certain that the data set that we're going to get from the platform is going to have two strings that have equal length. So if we're writing effective code, then one of the things that we want to make sure that we do is to ensure that those two strings have the same length. So we probably should make sure that we have statements in our code that say, well, if the length of S1 is not equal to the length of S2, here's where we want to ensure that we have an error message where we say that the two strings have unequal length. And generally, I don't have those types of statements, but that one seems to be so obvious to add that it makes sense to do so. However, a lot of the time, we're just going to be able to assume that Rosalind is giving us these data sets in the format that we like. But the idea is if we want to extend this code to other places, well, we need to make sure that it's a little bit more robust and protective against some of these errors. For now, though, I know that I just want to keep track of the number of transitions and the number of transversions that I encounter along the way. So I know that I will need one integer to be equal to starting out as 0, keeping track of the transitions, and another keeping track of these transversions. At the end of this process, if I think about where is it that I start, where is it that I end, I want to return the ratio between the two. So then it's just a matter of ranging over, now that I know that the strings have equal lengths, otherwise I would have produced an error. I just range over the strings, or range over one string. I can pick S1, I can pick S2, it doesn't matter. And keep track of which mutations are which. So I know that I'm going to have a loop that says for I in the range over the length of, I could do length of S1, I could do length of S2, does not matter because they're the same. And now I'll say, do I see a mismatch? If I see a mismatch, then I'm going to categorize it. So if it's the case that S1 of I is not equal to S2 of I, then I want to categorize. And here there's two ways of doing this. Number one, I could try to directly categorize which particular type of mutation that it is. And then I'm going to have a bunch of code that's inside of a for loop and then inside of an if statement within that. So if you've seen how I'd like to teach programming in general, or think through solving problems, which is really what programming is all about, I like partitioning out the amount of things that I can hold in my brain at any one point in time. So here's where I'm going to say, is it the case that this particular mutation is a transition? And I will pass all of that work to a subroutine. So that subroutine is simply going to take two symbols. It's going to take the ith symbol of S1 and the ith symbol of S2 as inputs, and it's going to return a Boolean variable or value. That Boolean value will be true if it is a transition, and it will be false otherwise. So in this particular case, I'll add one to the category, to this count transitions. Otherwise, I'll add one to the transversions count. And, technically, I probably should ensure somewhere that, well, this is a DNA string. So you might imagine I could also write a function and maybe place it here if I were being very precise. And this function would be called something like assert valid DNA string. And it would take S1 and S2, or it would just take S1, and I would do the exact same thing for S2. And I would have a helper subroutine in each of these cases that would be called to make sure that I actually have strings of A's, C's, G's, and T's, because if I have something else in there, like an X, well, this is going to be weird. I'm going to be adding one to the transversion count. But really what I care about is making sure that that's not the case to begin with. So it would be here where it's probably the most appropriate to make sure that we have that check. So again, sort of good engineering practice to make sure that you're protecting against these circumstances. And we could write that subroutine and make sure that we call it. Again, in this case, we know that we have a valid DNA string because we're only applying this to data sets that come from the Rosalind platform. In the meantime, I should write my subroutine is transition. And this is an example of a function that's going to have, actually quite a few possible cases. It's taking two strings, and I know that I'm only calling this function on strings of length 1. So one thing that you might want to do is, validate that both A and B have length equal to 1. So if length of A is not equal to 1 or length of B is not equal to 1, this is not going to make a whole lot of sense. So I'm going to raise a value error and say invalid lengths. Strings should have length 1. And I'd say that's one of the downsides of working with Python, where you don't have an explicit symbol type. And so everything is a string, which is useful, except for the fact that then when you write good code, you have to make a lot of these checks like this. But I know that I'm only going to pass strings of length 1 into this function, so everything's going to be okay there. And now here's where we say, I'm going to have a lot of cases, right? You could imagine you're going to have a bunch of switch cases where I'm going to have to say, like, if A is equal to A, and then I'm going to have a bunch of cases, and then I'll say, else if A is equal to C, and then cases, and so forth. And we could write code that way. It's not going to be that long, but I did want to show a shorter way of writing this function. And the shorter way of writing this function is to say, well, I either want it to be the case that both of the symbols are purines, or both of the symbols are pyrimidines, and I don't mind which of those cases is true, but as long as they are both purines or both pyrimidines, then I'm okay. But there's one other important thing, which is I want them to also not be equal to each other. So I think, there's an important point, which is I want to make sure that A and B are not equal to each other, which I know is not going to be the case by how I've called this function. But I also feel like, we want our code to be reusable wherever possible. This is a reasonable function. And in this particular case, when I call this, I know that the two strings are not equal. The two symbols do not match. But it would be weird if I write a function where I'm classifying a C to C mutation as a transition. That's not a transition. It's just two symbols that match. So there are kind of three tests that are going on here. I want to return true, if both of these things are purines, or both of these things are pyrimidines, then I want to return true. But I also want to ensure that it's the case that also A is not equal to B. So we have a nice little, this is a very pretty, I think, way of practicing some Boolean logic and some of our connectors, which is, I've not actually defined either of these variables yet, but the idea is if either of these variables winds up true, that's good. And then I also want to ensure that the two symbols mismatch. So if that's the case, I want to return true. Otherwise, I want to return false. So I'm kind of working backwards here. And before I even continue with figuring out what these variables are going to be, I will show you that, this way of writing code is so common that in practice, if you think about what is this expression, this is going to be a Boolean value. So this whole thing is either going to be true or false. And it's going to be true precisely when we want to return true, and it's going to be false precisely when we want it to return false. So in practice, you don't usually write code that looks like this, even though this is probably very intuitive. Probably the way that you would write this is instead, well, just return the value that you get from that immediately. Now, what's nice about that is we know what we want this to look like. The only thing that I have to do is figure out how to declare these two variables, both purines and both pyrimidines. But here is where we also can use a little bit of Boolean logic and say, well, both purines is going to be equal to something. Both pyrimidines is equal to something. And they're both purines if they're both either an A or a G. And we have a quick test in Python to know, is something a symbol belonging to a collection of possible symbols? And that is just by taking our symbol that we care about and saying, is it in the string consisting of the symbols that we care about? So ANAG, if the symbol A that we've plugged in here is an A or a G, then it will be in this string. So we want to make sure that this is true, but we also, for both of them to be purines, we're going to have an and then we're going to have the same check for B. Then for the other side of things, I'm going to do the exact same structure, but then I'm also going to replace the AG here with the CT here. So we wind up with a very nice, short, tight function that I think is very intuitive. It's almost, in a sense, it's almost itself very modular, and it's not truly calling a subroutine, but it's using the notion that we're declaring this Boolean variable here, this Boolean variable here, and then if either of these things is true, then we want to return true. Of course, we have this additional check that A is not equal to B. And that's it. So I can run Python 3 or Python on a Windows machine. And this is tran.py. And now I see that I believe I'm matching what we have on the website. So I'm going to go ahead and comment out my parsers. And I'm going to go to the website and get a data set. And put this thing here and run it. And now I get a value. That's going into output as well, but I could just copy it from the console and take it back to my website. Copy in and submit. And we got it right. All right. All good. If you've got any questions, let me know. Felix, you said you always have to remind yourself by saying purines are pure energy, so they make ATP and GTP. That sounds like the reasoning of a biochemist to me. With me, I would need a mathematical mnemonic of some kind. We're going to go to SSEQ is going to be our next one. It's a nice little warm up, but all right. Yeah, sorry, that was easy to check. It's the cool thing, Felix, is, I don't know, being in an interdisciplinary field. It is, I guess, technically we're transdisciplinary now, whatever that means, but it's like we're big enough, even though we're interdisciplinary. Comp Bio is big enough to be like its own field, but of course it's still pulling from all these different places. So one of the neat things is seeing how different people reason through the same question, looking at it from different lenses. And that's the neat thing about, I don't know, teaching students, when I get to teach students who have like a heavy biology background or a heavy computational background, and they come in and they see the same problem. They see it from like wildly different perspectives. For me, I'm just like, if I have to memorize something, I'm really not happy about it. Can always look it up. If the internet breaks, well. Okay. Next one. SSEQ. Now we've seen problems where we've looked at substrings of other strings. And this problem is going to introduce subsequences, which don't need to necessarily be contiguous. What this problem is doing is, am I particularly interested in finding a subsequence in another string? Is there a biological application for that? Not really, no. But what it's preparing us to do is to take two strings and then try to find a longest common subsequence of the two strings. So, in other words, can we find a sequence of symbols that doesn't necessarily occur as a substring, but that occurs in the same order in two strings? That is a fundamental problem. Like if you only do computer science, as a field of study, at some point in time, You're going to take an algorithms course where you see that problem, and you will solve that problem from a purely computer science perspective. But that is an unbelievably important question in biology because it prepares us for aligning two strings. If we want to find a longest common subsequence of two strings, that's equivalent to aligning two strings, allowing ourselves to have some insertions and deletions, and then finding the positions in those strings where we have matches. So if you're trying to align two strings to make them as similar as possible, the first thing you might do is to try and line up all those symbols and get all the matches that you possibly can. So that's an example of a question that is like a pure theoretical computer science question that you wouldn't have to learn any biology to be able to address. But then on the comp bio side, we love questions like that because they introduce us to more advanced dynamic programming. They help us do sequence alignment. And we will introduce those problems soon. But for now, we have a problem that tells us we have two DNA strings, and we're getting them here in FASTA format. Presumably, the first one is going to be longer than the second. And then we're trying to find a collection of positions or indices in the first string where these symbols in the second string appear as a subsequence in order. And there's lots of answers here, so you can return any one. Note, GTA occurs here. And notice Rosalind is 1 indexed, so we could just return 3, 4, 5. But the sample output is different from that to point out that in this particular case, we're going to return 3, and then 8, and then 10. Or we could have returned 3 and 4, and 10. So there are multiple different possible solutions. This problem is just getting you to see, well, return any one. So for the solution to this question, we have a function called find subsequence indices. And, the assumption is that t is less than s, meaning the length of t, I should say, is less than or equal to the length of s. And that would be a good thing to assert and make sure that we check. But the point with this problem is I'm looking for just one solution. So what I will do is I will look simply for the first solution that I find. So I want to keep track of the positions where I find this match, and I'm going to call those indices. And these indices are going to be a list of integers, of course, and they're going to start off as an empty list because I might not find anything. Then I want to keep track of the current index that I'm ranging through in the shorter string. So I'm going to use a variable j and set it equal to 0 to do that. And then the idea is range over the longer string. And what are we going to do? Well, we're going to see if we find a match with the current index of the shorter string. If so, increase j by 1 and log this index in indices. Okay, so once I've got that idea, I can see the process. I'm ranging over the longer string. I have a current symbol I'm trying to match in the longer string. If that current symbol ever does match, good. I'm going to move over one in the shorter string. So I'm going to move to the next symbol that I'm looking for. And I'm also going to remember that this match happened. So that match index is going to go into the indices. So I'll say, let's range an I over the longer string, which here is S. And here is where I want to check, is it the case that S of I is equal to T of J? So J is keeping track of my current index in the shorter string. I is keeping track of this current index in the longer string. And if that's the case, then I want to make sure that I append that index. And what am I appending? It's the current index of S. And I also want to move J over 1. So slide our current index in T over. As a result of this process, after you have done this, you know, for the entire string, it might be the case that you didn't use all the symbols of T. So we want to be careful with that case. It's very tempting to, of course, just say, I want to return the indices that I found. But I want you to imagine a case where, I don't know, you know, e.g., S is equal to a bunch of C's and T is equal to AA. Or maybe to be a little less cruel, let's say CA. So in that particular case, I definitely would have found a match with the C, but I never found a match with the A. And so what happens as a result of this is, well, I ranged over the entire string S, but then at the end of that process, I hadn't actually used all of the symbols of T. So I don't want to return the indices because I did not find a hit. In that particular case, what I want to return is not an error, but just return an empty collection of indices, not a partially filled out collection of indices, which is what this would return. It would return the index 0. So that would not be correct. And so to make sure that that's the case, and this is the benefit of kind of moving towards programming where we don't just have one randomly generated test, but we have a bunch of tests that you would run your code on. But if that's the case, the question is how to test it. And in this particular case, well, I know that I'm not at the end of the string and I've not considered every possible symbol and matched something. If it's the case that at the end of that process, that variable j that I created in order to store the current index of the string, if that variable j has not actually hit the end of the string yet. So if that's the case, then I want to return an empty list. There's one other thing too, though, which is I've not actually handled what happens if j becomes equal to the length of t. So as the result of this process, one thing that we might do is to ensure that, well, once j becomes equal to the length of t, I probably want to stop. I don't want to carry on with continuing to try to find matches, because what will happen is I'm going to exhaust all of the symbols of t when I'm matching it against the string s. So for example, in our case, we know we're going to get a hit for our sample data set. So if we look at this particular example, I know what this is going to return. It's actually not going to return 3, 8, 10. I think for our code, it probably should return 3, 4, 5. And once I hit here, well, then the question becomes, I could do one of two things. Probably the most efficient thing to do would be to say, if it's ever the case that the length of your indices becomes equal to the length of t, I'm going to stop the process and return the indices. So that's one way I could do this. Another way I could do it would just be to say, you know, I only want to continue making these checks of s of I equal to t of j for as long as the current index of t that I'm looking at is less than the length of t. So I only actually want to do this in this particular case. Either way is going to work. I actually kind of like the other way just as much, because to me that's a little bit more efficient. Like, we make a check to say if it ever becomes the case that the indices, you know, we could also do if the length of the indices is equal to the length of t, you could just return the indices. That's another way of handling this. So I can do it this way, or I can do it this way. Doesn't matter. Pick one. They're both going to be good. And in the meantime, I should be able to run this. So we will run with Python 3 SSEQ.py. And as I said, you know, this says 3, 8, 10 is expected. Well, we get 3, 4, 5, but that's correct too. So the autograder is going to be able to check either of these submissions, I hope. And let's do that. Let's comment out the parsing part of our code. Let's get a data set. And let's put that over in our folder. Call it data set. Run it again. And now we can look at output. copy in all of our output. That's given us the subsequent starting positions. And we see that we get it right. All good. Hooray. If you've got any questions, as always, just let me know. In the meantime, I'll get set up on our next question, which is GRPH. All right. GRPH is going to introduce graph theory, which is the study of networks. And that's probably a good thing, because in biology, networks are everywhere. What are networks? Networks are representations of objects that also have pairwise relationships between them. So in biology, you see these sorts of relationships all the time. For example, you might imagine a network where all of your nodes are proteins, and then we connect to nodes in that network with an edge if there is a known interaction between these two proteins. So, and studying those networks on their own is an entire field of study, right? To sort of make predictions. If you might have some information about some of the proteins, but not others, how is it that we could make automatic predictions based off of network structures in order to figure out what, for example, the functions or the associated behaviors of some of these proteins are that are not studied? Or your network might be a collection of all proteins again, but maybe some of your proteins are transcription factors, meaning they regulate other genes. And so your nodes are proteins, but you connect two nodes with an edge if protein 1 is a transcription factor that regulates the expression of protein 2. So there's many different ways in which networks are very useful in biology. Probably the most prevalent example of a biologically inspired network is a neural network, which is a way of looking at our own nervous system and trying to borrow a model from it, where neurons become our nodes and connections between those neurons become edges. So in that sense, a biological network is very much changing the world. This network that we're going to work with in this particular problem might seem a little bit strange. In this particular case, the nodes are going to be represented as strings. So we have a collection of strings, and we're also given some integer k. And all of the nodes are going to be represented as strings. All of the edges are going to be represented as, do these two strings overlap in a sufficient number of positions? The problem here probably should have k as an input parameter. We've hard-coded k equal to 3 for this problem. So we're just giving a collection of DNA strings. And to understand how this network is going to work, you might say, well, what this says is I'm not going to allow connections from nodes to themselves. So technically, this string overlaps with itself because the last 3 symbols here are equal to its first 3 symbols. That's useful. We're not going to add that to this particular problem. So we're looking only at edges between distinct strings. But if you look at this string here, notice that the final 3 symbols here match the final three symbols of this string. So we would have an edge then from this node connected to this node, but not in the reverse direction. So our edges are going to be one directional. It's not the case that the last three symbols here match the first three symbols there. So at the end of the day, we're going to have what's called an adjacency list, and all the adjacency list says is, in this particular case, the first label here indicates the first node in the edge, and the second label indicates the second node in the edge. So we said, we're going to have an edge connecting this string to this string. So this is the corresponding adjacency list element for it. Of course, we're also going to have that this string overlaps with this string. So you'll see that corresponds to the second element in our adjacency list. And then the final three symbols here in 2391 match the final three symbols in 2323. Okay. So there's our final element. And you can check that there are no additional overlaps of three symbols that you see between the strings. This problem might be a little weird in that the network might seem strange. What's the interest biologically in such a network? And it turns out that when we solve the problem of genome assembly classically, and this is a problem that goes back to a Nobel Prize winning sequencing of a bacteriophage in the late 70s, the picture has always been the same, that you may have a genome with 5 billion nucleotides in it, but at the end of the day, we sequence genomes by capturing many copies of those underlying genomes, say with a blood sample, and then isolating white blood cells, and then fragmenting down the many copies of the genome into short fragments of DNA that can get fed into a sequencer. And then we need to use overlaps between fragments that we sequence in order to try and lay out and figure out what the underlying genome is. So this network is very useful in that endeavor. And the reason why is because it allows us to pre-organize all of our data into a data structure that helps us figure out, which pairs of fragments of DNA actually overlap. So in this particular case, if you knew that this overlapped only with this, you could start building a string by adding on the TTTT to the end of this string. And then this overlaps with this, so you're going to add CCC to the end of that string. That's not what this problem is about, but it's what this problem is building towards. And so we will see that in a later problem. For now, though, I just want to return this adjacency list. And notice the adjacency list is with respect to the IDs. So what the data set is going to contain is a collection of tuples or pairs. You'll have the ID associated with this particular string, and then you'll have the string itself. So when we move towards the solution, the overlap graph function that we're going to write to solve this problem is going to take two things. And even though K is hard-coded into the data sets that we're going to get, I'm going to solve this problem for an arbitrary value of K because that's the stronger way of doing this. But our sequences, as I said, are going to be a collection, a list of tuples. Each tuple is going to contain the sequence ID as well as the string itself. At the end of this process, I'm going to return also a collection of tuples. So now the tuples that I'm going to have are going to be sequence IDs in both cases. So what the tuples will represent is to say this is the first node and it's connected to this node. So I'm going to have each one of these tuples represents a single edge in this overlap graph that I'm producing. The idea there is really taking, the data that we're given, and after we make sure that we parse it out, understanding that each data point corresponds to these two things. So for our example, notice that the sequences are a list of the 5 tuples that we saw previously, but we've separated out every individual element as containing the ID of the sequence and its string. So that's what we feed into overlap graph. And what I want to obtain from this is a collection of edges. So that's probably what I should call the thing that I want to return. And the thing that I want to return is, as we know, it's a list where each element is a tuple of two strings, representing the IDs of the things that are connected via an edge. It starts out as empty. At the end, that's what I will return. And in the meantime, the idea is, well, range over all of the sequences and strings and try to find a overlap in all the other sequences. So we're headed for a double nested for loop. On the outside, I'm going to say, well, I care about the label of the string. I'm going to call this string S. And I care about the sequence. And in biology, we talk about this, a sequence just meaning a string. So there is, you know, maybe we could have said biological strings or something, but the notion of a sequence often just means a DNA string, an RNA string, or a string of amino acids representing a peptide or a protein. So we're going to use the term seq to really just mean, or sequence just to mean strings. So sequences here is our list of tuples. So I'm going to range over that list, and I'm going to obtain a tuple as the current element when I range over it. Then what I want to do is I want to say, well, what is it that I want to match? I'm interested in the suffix of the current string. So I'm going to look at seq s, which is my current string. And then I want this suffix to have length k. And here's a classic string indexing question. So you would get very comfortable with this. The way that I would want to have you think about indexing a suffix is to say, well, I know that there are two elements, there are two indices when I specify what this substring is. If it's a suffix, the second index is going to just be the length of your current string. So note, all these strings could have varying lengths, but the current string that I'm working with, we're going to say we know that it's going to end, its final symbol is length of seq of s minus 1. And that's not included when we do substring indexing. So we always know that the final element here, if we're looking at a suffix, is just going to be the length of the string. Then we use a helpful tip. And my helpful tip always for substring indexing is we easily know the length of my substring if I subtract the higher element minus the bottom element. So if I want a suffix of length k, well, I just need to make sure that when I subtract this minus whatever this is, I get k. And that's easy to do. I'm going to say length of seq of s minus k. Make this together. Because when you subtract this minus this, well, all that's remaining is a k. And the second index is not necessary. So in practice, you would just see this. But if you saw code that looked like this, it might not make the most sense to you. And I think it's far easier to understand as the final element, because the suffix is always just the length of the string. And this minus this needs to be equal to k. And so at the end of the day, yes, we can get comfortable and get rid of that. But on the way, it allows us to understand it a little bit better. Now what we want to do is range over all the other strings, grab their prefixes, and ask, you know, does the current prefix match this suffix? So I'm going to have another for loop. I'm going to range over the labels, but I'm going to call this label of t. And I'm going to range over all of the strings, but I'm going to call that seq of t. And I'm going to range back over all of my sequences. And then I'm going to say, all right, the one thing I want to make sure is, you know, don't check a string against itself. So if it's the case that the label of t is not equal to the label of s, the other thing I might do is, well, what would happen if you have duplicated sequences? So maybe it's the case that seq of t is equal to seq of s, so maybe I should do it that way. The problem doesn't really specify it, and I think it's clear from the problem that we're not going to see such a case, so it's all right to write it this way, but you might imagine it in the other way too. So don't check these strings against themselves. In this particular case, I'll say that the prefix that I care about is equal to, well, this one I think is easier. I'm going to look at seq of t, and I'm going to go 0 to k. So I know that my first index needs to be 0. I know that the difference between these needs to be k. So at the end of the day, well, what's the prefix going to be? It needs to start at 0 and go up to k, but not including k. Of course, again, in this case too, we rarely will write 0 as the first index in a substring. Probably we're just going to get rid of the 0. Now I say, do they match? So is it the case that the prefix is equal to the suffix? And if so, well, let's append it. So what do I append? Well, the edges are going to store these connections, but the connections themselves are just a list of tuples where it's the label of the first string and then the label of the second string. So I'm appending label of S and label of T. Notice that there is double parentheses here. And the reason why is, well, I always have this notation for append. Okay. But in this particular case, I'm appending not two strings, I'm appending a tuple of strings. So we want to make sure that we have two collections of parentheses there. And that should solve the problem. So when I run this, I can go to Python 3. I can run grph.py. I see that it appears to be working on the sample. So I'll get a larger data set. And I'll put that data set into our folder. And I want to make sure that I comment out my parser and run. And take that output and put it back in. Hooray. So another problem down. On our stream, I think we have 3 down, 3 to go. Let us know if you have any questions. I'll go ahead and start getting set up. Our next problem is going to be K-mer. All right. Chris, you said, which label of the sequence am I calling a label? Is that a paradox? So, I mean, like if this is FASTA format, one of the nice things about Rosalind is it gets people comfortable of seeing this format because it's ubiquitous, right, in biological data analysis. But FASTA format always has a header describing this thing. And then it's got the string itself following that. So the header is indicated by this line that has the greater than symbol starting it. So this is nice because it means I'm not representing the adjacency list as string to string. I'm just grabbing the information that's in this header. and saying that that's the name of this string. So the adjacency list is going to do this. The idea being, well, presumably you could make a little dictionary behind the scenes too, so that you can remember what the identity of Rosalind 0498 is. You know, if you actually were going to use this network for the purposes of something like putting a genome back together, which we'll see soon. Which part of the sequence is the label? I'm confused. To me, it's just this. This is our ID. So the sequences themselves are pairs. You have, this is the first element and that's the second element, which when we looked at our code, if I go back to it, when we ranged over it, it's a problem we just solved, GRPH. We range over the sequences. The sequences are these pairs. So the label is the ID. Okay, good. Cool, cool. All right, good, good. We're going to solve problem K-mer. Make sure that I'm set up. Rodney, welcome from Kenya. Good to have you here. All right. We're now going to look at K-mer. And K-mer is a mean problem. We probably could have just asked you to take a string and chop it up into its K-mers. Now you might remember the term K-mer in computational biology is a very commonly used term. And all it means is a string of length k, often just a substring of length k. And so that's what this question is asking us to do. If we connect this to our previous question, well, imagine that you produce an algorithm that uses what we have seen with overlap networks to apply it to genome assembly. So you want to have a bunch of fragments of DNA and you want to start overlapping them to produce the underlying genome. One way that you could test your algorithm would be to take a string like this, chop it up into all of its K-mers, and ignore that you knew the original string, scramble them, and then try to put the string back together using all of its substrings of length k. So you might get this string, and then you might get this string, you might get this string, and so on. And when you scramble them, you're not going to know what the correct order is. So the question is, could you put them back together? So the idea of working with the collection of K-mers of a string is really important in this particular application. So what the question is basically asking is to chop a string up into K-mers. But I think our sense was, can we do something a little bit more fun and funky? So instead, what we're asking for here is not just the collection of substrings of length k from this string, which at this point I think would be a boring question. We just slide a window along the string and append each K-mer that we see to a growing list. What this question is going to say is, all right, first, what we want you to do, so you're going to see a big array. What we want you to do is order all of the possible K-mers lexicographically first. And then we're going to count how much each K-mer occurs in the string, and then we're going to log it in the associated values down here. So when you see these values 4, 1, 4, 3, first off, we've hard-coded the parameter k equal to 4. So we probably should have a function that works for an arbitrary version of k. That's what I'm going to write. But when we apply it to a data set, k is just going to be equal to 4. But when you see this 4, 1, 4, 3 in this longer array, well, first off, the length of the array is equal to 4 to the 4th, because that's the number of strings from the DNA alphabet of length 4. You have 4 choices for the first one, 4 for the second one, 4 for the third one, 4 for the fourth one. So we just wind up with 4 to the fourth, which is just 16 squared, or 256. And so we've got all of those values. And now we have these values. But what does 4143 mean? Well, this 4 corresponds to AAAA. This one corresponds to AAAC. This is AAAG. And this is AAAT. So the idea is, if we give you a string, can we log the number of occurrences of all of those? Now, pick your favorite of those. I'm going to point out, notice AAAG occurs there. It occurs here also. So you see we're accumulating these occurrences of AAAG. I also see some of our AAAs. I see all of them here. There's one here. Here's a second one. And notice here we see 2 occurrences because we're considering overlapping occurrences. So we get that position, but we also get that position. So that's how we get that value of 4 in the sample output. This is going to seem challenging because I have to do a lot of work, right? I need to range over the string and identify, for the current K-mer, I want to add 1 to something corresponding to that K-mer, but I've got to go through and identify exactly where that K-mer corresponds in this table. There's kind of 2 ways of doing that. I guess number 1, I could, in real time as I go through, say I've got the symbol, the word ACTC. Can I convert that to a number, which will be an index over here? We actually have a little bit of hope for that, I would say. For example, if you think about AAAA, the A's are going to represent like zeros. So if we convert AAAA into zeros, then this would be the element 0. And this is AAAC, so C is going to represent 1, G is going to represent 2, and T is going to represent 3. But then you come to here, and what's this? This is going to be AACA. So that's at element 4. And the reason why we're getting that is, well, now we've got to convert, how would I know that that's at position 4? The reason why is AACA, if I'm thinking about my numbers as A equal to 0 and C equal to 1, now that C is equal to 1, but what I'm representing here is base 4 notation, where A is 0, C is 1, G is 2, and T is 3. So when I see AACA, that's really the number 0010 in base 4, and that's the number that we think of as 4 because it's saying when I see 0010, that 1 that I see is in the place of the 4 to the 1 column. Now, the problem with I would love to go about doing the problem that way because I love doing math in exotic bases. A lot of people, I think, are not going to love that. And the question becomes, well, can we get at this problem in a simpler fashion? Really what I want to do is figure out what the order of all these 4 to the k K-mers should be. And once I have that, I can just range over the input string, find the current K-mer. So the current K-mer is CCGA. And instead of doing a bunch of fancy math to figure out where this CCGA is, just kind of put it in the appropriate place because I pre-computed what the order of the K-mer should be. So instead of doing the fancy math, I would just look up CCGA in a table that tells me where I should send that CCGA somewhere in the middle of this table. So that's the difference between kind of a beautiful math solution and a clunky, inelegant computer science solution. But we're going to take the clunky computer science solution because although my memory is pretty bad, it's not so bad that I don't remember that we've actually already solved the question of generating all of the possible K-mers lexicographically in a problem that we've already solved. So LEXF was a problem where, and here's the benefit of having doc strings because I can go back and see it. And we solved this problem recursively, which was really cool. But I'm given an alphabet, and I'm given the length of the strings to generate. And what this function returns is the length of all K-mers from that alphabet in lexicographic order. Great. So now I'm just going to say, I don't need no fancy math. I'm just going to say from lexf, I'm going to import the function that I care about, which says enumerate K-mers. And now I have, that's most of the engine of what my function is going to be, is taking this function that I've already written and using it as a building block for this problem that without it would be challenging because we'd have to write that function from scratch again. So our idea then is to import that function, and then we're going to build a dictionary that's going to map each K-mer to the number of times that I see it. The first thing, of course, I want to do is to get all the K-mers. So I get all the K-mers in lexicographic order by calling our subroutine. Remember, when we look at the subroutine, it takes an alphabet as a list of strings. Okay, fair enough. I know what my alphabet's going to be. And it's going to take the value of K that I care about. That value of K is already a parameter here. So I'm going to say enumerate the K-mers. And now, again, it takes the list of strings as the alphabet. So it's going to take A, C, G, and T. What's nice about that is I can scramble the order, and I'll just have a different ordering of the alphabet. And this function will work in that case too. But I also need to give it a value of K. And now I've got that collection of all possible K-mers in order. Well, we want to build up a dictionary mapping each K-mer to the number of times that occurs. So I'm going to call that counts. It's going to be a dictionary. It's going to map strings to integers. And it's going to start off as being empty. And then I want to say, well, range over all of your K-mers and set counts of K-mer equal to 0. And so a lot of these K-mers that I'm going to have are never going to actually get updated, but I want to make sure that they all start off as equal to 0. Here the assumption is I'm only applying this function for small values of K because I'm going to be building something that's of size 4 to the K. So if I start making, 4 to the 50, I'm going to have an enormous dictionary that's too big to even store. So presumably I would wind up with just a bunch of zeros in that. But I'm going to set everything to start off to be equal to 0. And then what I want to do is, let's range over all the K-mers of the text and add 1 to the current element's value in the dictionary. Well, that's second nature at this point. So we're going to have 4i in range. The question is what to range over. And this goes back to when we looked at substrings of a string. Every computational biologist should probably have something akin to a tattoo on their body somewhere that says, I always know that if I want to range over the starting positions for possible K-mers, well, I know what that range should be because I know how many K-mers there are. I know that there are, in a string of certain length, if I'm looking for the strings of length K, there are length of S minus K plus 1 total. And the starting positions are going to range from 0 up to, but not including, length of S minus K plus 1. So that's going to ensure, when we do this ranging, it's always going to be this way if I want the starting positions of all of my K-mers of a string. Then once I've done that, I'm going to say, what's my current K-mer? Well, it's equal to, I don't even need this, but it makes things nice and clear. My current K-mer is the substring of S, starting at position I, having length K. So when you subtract the top minus the bottom, you're going to see I get K. And then we say, look at your counts dictionary, access the K-mer, and add 1 to its value. And at this point in time, think, what is it that we have, right? We've done a lot of good work. We have the list of K-mers. But we also have a dictionary mapping each K-mer to its number of occurrences. So we kind of have all of the values. I guess we kind of have all of the values that we care about. Let's look at what I mean. We've got the list of all the K-mers, and we've got them in order, and we have the values that I care about. But the problem is, if I just range through the dictionary and grab all these values, they're not going to come out in the order that I care about. So these are the values of the dictionary that I currently have, this counts dictionary. I've got that, but I want to make sure that I get all of these values in the order that I want, which is the lexicographic order of the underlying substrings. So I want to get AAAA first, I want to get AAAC, then AAAG, then AAAT, and so on. So when I do that, it's not just a matter of ranging over the dictionary. I want to say, what is it that I want? Well, I want a list of integers at the end of the day. It's going to start off as empty. That's going to be what we return. But in the meantime, I want to have a ranging that goes through all K-mers, because I know that if I want to access those integers in the correct order, the correct order is given by the order, this sorted order in all K-mers. So I've already figured that out, right? I don't have to sort anything. They're already sorted. And so since they're already sorted, I can say range through them, and then what is it I want to append to the composition? The current element that I care about is just counts of K-mer. Okay. And the other thing I could do is, I don't have to start this as empty. I could also start my list to be equal to, to have the appropriate length. So it could be, 0 times the length of all K-mers. And then I could just say, like I and K-mer over this, and then set composition of I. In Python, often we don't do things that way. In other languages, we might write code that way. In Python, a lot of the times we're just going to say, look, start this as empty and append as you go, and that's reasonably efficient enough. So now we've written our function. It's way less mean than it seemed. And the reason why is because as we go through the platform, we're going to be able to use problems that we've solved as building blocks for others. Okay, I'm calling the wrong file. So let's say python3 kmer.py. And here's our example. Do we see 4143 at the start? We do. So I should feel pretty good that I can comment out my parser, or uncomment out my parser, I should say. Get a data set, put it in the right place. We're going to make sure that we name it dataset.txt. And then I'm going to run my code one more time. That's pretty quick. So it goes to output. It gives me all of these values. So this string was very long. It appears that a lot of things were identified a lot of times. And the balloons and confetti come down upon my head. So we'll see that a lot as we go through. A problem will be very hard, but as long as we understand which problem or problems it's built upon, we can presumably use those as modules to build up a larger solution. That might be the most compelling example that we've seen of that thus far. If you're interested, of course, try solving this problem in the other way. Go learn some base 4 mathematics. Assign A equal to 0, C equal to 1, G equal to 2, and T equal to 3, and then you can range over this thing. You can just start making this array directly by saying, well, if I see ACAG, G is 2, so I'm going to say this is 2. A is 0, so it's 0 times 4 to the 1. C is 1 times 4 to the 2, which is 16. And A is 0 again. So this ACAG is 16 plus 2, or element 18, wherever that is. So you would just go in and you would add one to the element corresponding to index 18. So Ronnie, good question. I mean, this is kind of a toy problem. The problem that we really care about is going to be, for this particular application, take a bunch of K-mers, not represented as this table, but just take a bunch of K-mers and then try and assemble them into a genome that contains all of those K-mers that's as short as possible. So use overlaps between your strings and then make a long genome. If you do that, then sure, you'll probably shuffle them. And that way you can validate your algorithm by saying, I took a bunch of, randomly generated data and then I shuffled it to ensure that I don't know the correct order. All right. Next problem is going to be LCSM. Two more problems to go. We're going to do LCSM, then we're going to do KMP. With LCSM, or finding a shared motif, we've talked a little bit about substrings and subsequences, but a lot of computational biology and bioinformatics is about finding shared things. We're going to start here with the problem of a shared substring of multiple strings. We're also going to look at a shared subsequence of two strings. And in either of those cases, if you're following parsimony or you're looking for a pattern, well, presumably what you're looking for is something that's as long as possible. So we're heading in the direction of more advanced analyses of strings and finding things that are hidden within strings. In this problem, just for saying, I've got a collection of input strings, and then I'm looking for a substring of those strings that's as long as possible. So that substring should be shared by all of these strings. And there might be multiple solutions, in which case the autograder is going to be fine with any one of them. But in this case, the longest possible substring you can get has length 2, and you can see the occurrence of AC here, and AC here. For the solution to this question, our idea is going to say, well, let's figure out what the shortest string is first. And the reason why is the longest common substring of all the sequences, in the extreme case, it can never be longer than what the shortest string is. Imagine that all of your strings are just the letter A repeated. Well, then what the answer there would be the length of the shortest string. So the first thing we'll do is identify the shortest string. So I will set what I will call shortest equal to sequences of 0. Sequences here is my input list of strings. And in this particular problem, I care only about the strings. I'm not interested in any particular ID of those strings because I'm just going to return a substring corresponding to something that's shared with all of them. Once I've got that shortest string, I'll say, let's range over the sequences. Or once I've set that first string, let's range over the sequences and find, can we find something shorter? So if it's the case that the length of the current sequence is less than the length of the shortest thing that we've found thus far, well, then I want to update the shortest string to be equal to the sequence. And Python gives us like one-line ways of writing that. From my perspective, this is the easiest way of understanding exactly what's going on. I'm just ranging over the strings and figuring out which one is the shortest. Now, once I've reached this point, there are two directions we could go. And we're going to apply brute force. So I'm just going to range over all the possible substrings here and then say, what is it that I find? Direction one would be to say, let's look at, I should say, we will look at all substrings of shortest. And when I look through all these substrings, what I want to find is the substring of shortest that's contained in all the other strings. One direction I could go with solving that problem is to say, all right, let's try small strings first. And if you do that, well, you need to range over all possible sizes of strings up to the length of the shortest string that you found. The last thing that you find is the answer. There's a second approach, which is the direction that we are going to go, which is to try big substrings first. So this is range over all possible sizes of strings, starting with the length of shortest, moving downward. The first shared substring that you find, stop and return it. That's nicer, right? Because then I only need to look for one thing. So instead of ranging over every possible substring length, if I find a long substring, I want to just stop the algorithm. That might mean that we have to do the ranging in a slightly more exotic way, but fortunately Python is going to give us, the way to do this. So we will apply idea number 2. And in particular, I know how to range over all of the substrings of the data set. I'm going to have a variable called length. And if I were ranging upward, I would say, well, I'm ranging over the length of the shortest. And what am I saying here? Let's say n is equal to the length of the shortest. I like doing that. So we're going to say I'm ranging over everything up to, but not including n. And that's idea number 1. But idea number 2 would be, let's start our ranging of length at n. And I'm going to go down towards 0. And I'm going to decrease by 1 each time. So Python gives us this step size, which is allowed to be negative. And this allows us to start at big values and go down. So that's going to be how we apply idea number 2. And now we say, well, what are the possible substrings? Range over all possible starting positions in the shortest string. So I will say, for all of the start positions that you possibly could imagine in a range over, well, here's where we get to apply. Length is now my K-mer length. So I know how many substrings there are going to be. There's going to be n minus length plus 1. So that's our magic formula in computational biology that says if my string has length n, and I'm looking for all substrings of length, given length, the starting positions of those strings are going to range between 0 and n minus length plus 1, not including n minus length plus 1. Of course, we don't need to specify 0. Now we say, what is the string that I care about? The candidate string that may or may not occur in all of these strings is equal to the shortest string and indexed starting at start and ending at start plus length. Okay, this is the starting position. You subtract the top index minus the bottom index, you get the length that you care about. And now I want to say, does it occur in all strings? To this point, I will say if, and now we're going to use a subroutine. I don't want to continue thinking I'm 2 levels down in, I already have a double nested for loop. So instead of doing this, let's say if it's shared by all, we're looking for the candidate in this list of all these strings. If that's the case, then I want to return the candidate. So this ends the algorithm. Is now over. I probably need to make sure that I do have a default parameter, but if I make it here, I didn't find a single string shared by all. Okay, that's tough, especially if they're from the same alphabet. You'd hope that they at least include a shared symbol. And Rosalind's probably not going to check us on this, but that's another example of where good code would be able to make sure it handles this particular edge case. In this case, I found a hit. So I just need to write this subroutine, and I could make it a little bit tighter in the sense that I know that the candidate is contained in shortest. So I technically only need to consider whether this candidate substring is contained in all the other sequences and not in shortest. I'm going to be doing a little bit of redundant work. But you imagine if you have a bunch of strings here, you have 20 input strings. And if I'm checking it against 19, I may as well just check it against the 20th. So I could optimize this a little bit just by remembering which of these strings is the shortest, or rearranging the sequences so that the shortest occurs first. And then I would only need to arrange over like sequences of everything past the initial element. But I have not made that assumption. So because that's the case, I'm just going to have a little bit clunkier, a little bit more redundant of a function, but it's still going to be short in terms of longest common substring. And now shared by all is still just going to take a substring and a list of strings, and it's going to say, does this substring occur in all of my substrings? And this is now an easy function to write because this is the type of thing that we've seen in much earlier problems. So we should be comfortable with a function like this because I'm just going to range over all the sequences and ask if it occurs. And here's where I could do one of two things. I could go back and call a pattern matching subroutine that I've written in a previous function. That's great. So I could use my own code. At this point, I think it's fair to say we also can just use a little bit of built-in Python. There's nothing wrong with that. So I'll say for each sequence in the sequences, does it occur in the current string? And if it doesn't, we have a nice little quick check here. So we say if it's not in seek, meaning it's not a substring of seek, there's a nice Pythonic way of doing it. Well, I want to immediately return false because it's not contained in the current string. And the idea is if I survive all this, what do I know? I know that candidate is in all the strings. So I'm just going to return true. And we're going to check this on our sample data set. So I'm going to Python 3 LCSM.py. I get TA, I think, which is the point is that they all contain TA. So TA here, and TA there. That's another acceptable answer, so it's all right. We can return one single solution if there are multiple. Let's try our hand at downloading a data set, putting it into our folder, naming it dataset.txt, deleting our old data set, commenting out our parser, and running it again. We've got an answer here. Our data set was reasonably large, and this took, a couple seconds. So our input data set had a bunch of strings in it. Yet this algorithm was good enough to finish in a couple seconds. We'll copy in our solution. And we get it right. So a couple points there. Number one, I would encourage you to try this with our idea number one, meaning starting with short strings and building your way up. What you're going to see is it's going to be slower because after it hits the correct answer, it's then going to be trying long substrings that are not going to work. The question is, how much slower is it going to be? In this case, we found a pretty long substring. It wasn't that long, so you will continue having to identify shorter strings. But it's not going to be that too bad. However, it is not the fastest algorithm. If you were wanting to find shared substrings of a bunch of strings, you would probably want to construct something like a suffix tree or produce a data structure to organize your data in advance before you actually go through the data, because otherwise we're going through the data blind and we're just saying, can we find anything by brute force that we find to be matching? It's a good setup for our final problem of today, which is going to be called KMP. Right. KMP is the problem speeding up motif finding. And it's built upon a problem that we have already solved, which is SUBS. That SUBS problem is all about finding a substring of a longer string. There's a question here, which is, as we try to match a given string against a longer string, the classic way that we have solved this question is to say, well, slide a window along the longer string. And in each window, ask yourself, do these two strings match? So, does the current window of the longer string match the pattern that we're looking for? That's simple enough. And there's a brilliant idea, which is that, can we do better than this? So this is the subject of what's called the Knuth-Morris-Pratt algorithm. And their brilliant idea is that sometimes when you slide the window one symbol over, you actually already have a fair amount of information from the fact that you may have already tried to start matching symbols. And when in our algorithm, when you slide the window over, you ignore any of that information. So the example I think that we give in the introduction to the problem is, imagine that you have a pattern that you're looking for against this longer string. And you reach a point here where you started matching a lot of symbols. So if you're matching a lot of symbols, you're accumulating a lot of information. So we start matching ACG, TACG, and all of this matches. So we get 7 positions that match. But then as we do this, the next position that we encounter is a G. And so we don't actually obtain a match against T. That's a shame. We were off by 1 symbol. But along the way, we probably learned some things, right? So in particular, I know that the first symbol of T is A. And as we go through this, well, as I've consulted all of these symbols, and I know, for example, along the way that C, G, and T don't match this starting position A. So I could, in theory, remember all of that information so that I don't need to start the next window here. Why on earth would I do that? Because I have already seen that this is a C. There's no way it matches the A. So I should just be able to skip over the C, the G, and the T. So those should be easily skippable because they don't match here. Then the next question is, and notice how that this is a little bit of a special string because it's a repeated string, ACGT, twice. the reason why that's our example is when you hit this point in this window and you see that you failed at this string, this symbol G, do we really need to, what else do we need to consider? And so in this particular case, when we look at our next symbol, oh, I see ACG, that's really good, right? So I know that the AC and the G matched, or I could keep track of that as I go, but also I know that this symbol here is a G, which I obtained as a mismatch here, and I can see that it would have been a mismatch there. So the point of KMP is to do a little bit of work in advance to keep track of, to sort of index the string T in advance so that when you fail at a certain position, ACGT, ACGG, you can go ahead and know that there's no way that you need to proceed with this window and this window and this window and this window. And in particular, I know ACGG cannot be a match because I can see it here. So the clever idea is to do this. Now, the implementation of that idea is where things get really, really brilliant. And what this question is about the foundation of the algorithm, which is taking the shorter string. So there's a little bit of a typo, I would say, in this problem that I've noticed, which is that we're computing the failure array of the shorter string. So we call it S here, but what we really mean is the shorter string that I care about. So this is the pattern that I'm going to match against a longer string. What the failure array keeps track of, and I'm just going to solve this problem for now with producing the failure array of a given input string. So given the input string, we're supposed to return the failure array. The failure array itself is defined as, well, P of K, so it has length equal to the string that we're looking at. P of K is the length of the longest substring that ends at the position K that's equal to a prefix of length equal to that substring. So in this particular case, equal to the length of this substring. Okay, so what is the length of that longest substring? So in this particular case, well, in this case, we look at like this string, the symbol for example, it's an A. Okay, so there's nothing that ends in A that starts with C, so those things don't match. Same thing here. There's nothing that can end with G that's equal to a prefix over here, so that won't work. But in this particular case, there is a string that ends with the C that's equal to a prefix. It's this C, so we make a value of 1 there. And in this particular case, there is a string that ends in this A that's equal to a prefix of the string. Notice that it's here. The longest you can do is 2. And then you get a bunch of zeros because you have a bunch of things that don't match. But then if you look at a value like this, so you say, what is that value associated with that? That's also a CA. Okay, so the best you can do at this position is to say this CA is a match for this CA. Let's continue extending. So let's see what happens if you go all the way to here. So at this position, notice you have CAGCA, and that matches the CAGCA that you have here. So the idea then is to say, if I fail at this point, I know that I have failed against the first 5 symbols of the string. So if I'm matching it against, a longer string, and I hit this A, and I know that this A doesn't match, for example, I give a T in the underlying string. Well, I also get for free the fact that this starting string won't match either. So I don't need to consider that window because I would otherwise go 1, 2, 3, 4, 5 and hit a mismatch. So this is a clever idea. This problem is just going to be about producing the failure array of a given string. So to solve that problem, the problem is, remember, the failure array has P of K equal to the length of the longest suffix or prefix of the string that is also a suffix of the current string. So we're matching a prefix of everything up to this current point with a suffix of everything up to this current point. So it's a pure string question at this point in time. And if we want to implement it, well, the idea then is, we're going to first initialize the failure array. So I can practice that by saying failure is going to be a list of integers. That's what we're going to return, a list of integers. And that list of integers is equal to, well, let's start it off as a bunch of zeros, and they should have length n. Once I've set that, not non-local, okay. Once I've set that, then we're going to do a ranging. So the ranging that needs to happen is for k in a range over all the values that I want to set. So we'll go from 1 up to but not including n. So failure of 0 is going to be equal to 0. And then we range over the remaining values. And now we say, can I match a prefix of s of k to a suffix of s up to k? So I will use the same idea that we actually previously saw in LCSM, which is I could start with small values and try to match these prefixes building up. And then I need to consider all possible values. And the last one that I find is going to be my answer. But a smarter way of doing this is to start with the biggest imaginable prefix match that you could have with a suffix match, and then range backward. So I will have all the possible lengths in the range from k to 0 to -1. And I will say prefix is going to be equal to s of everything up to that length. The suffix is everything at the end of the string. So in this particular case, it would be s of k plus 1. Well, we're going to go up to k plus 1. So I should say this is k plus 1. K plus 1. So we'll go up to k plus 1. That's going to keep us from just matching the string against itself. In this particular case, k plus 1 is our final element. We want the suffix to have length, which means I'm going to say k plus 1, and then I'm just going to do minus length. Again, this magic way of thinking about indexing makes this so easy because I'm going to end at position k plus 1, at position k inclusively, but subtract the top minus the bottom and you get length, which is the length of the prefix. So we want these two things to have the appropriate equal lengths. And now I say, if it's the case that the prefix is equal to the suffix, then we're going to say failure of k is equal to length. And once I find that, I have set the failure value for k, and I should break the current for loop. So I'm going to say break. And that break will break this for loop, but it's going to carry on with ranging over all the values, all the possible values that I need to set in the failure list. So at the end of that process, I'll just return that list that I have. So this is the data structure that we care about to appreciate how this algorithm for faster pattern matching is going to work. What we would like to do, of course, is to, take this algorithm and compare it against a brute force approach and see, for these data sets, which one is faster. We, of course, have to write the algorithm to take this failure array and do the brilliant speed up that we have. So one thing I'm missing is n is length of s. But the point is we have not talked about the algorithm itself for pattern matching. The only thing that this question is asking us to do is to produce the failure array. So let's see how we do with that. So when we say, Python 3 kmp.py, you'll notice we're able to produce that failure array, and it looks, I hope, pretty similar to what we saw before. So we should feel good. Let's download a data set, and then we can put our data set into our folder, call a data set, go to KMP, make sure we comment out our parsing, run it again. We see that the sample is working here, but there's just one problem. The problem is that this is running behind the scenes, and Rosalind only has a 5-minute limit. And that 5-minute limit is there for a variety of reasons. One of the ways is to make sure that before you get a data set, you've written your parser, you've got everything ready to go, you know how everything needs to be set up. So all that you need to do is run your algorithm, run your code on the data set that we give you. I like that. It's a nice model, and make sure that you're prepared. This is, through the course of this series, the first time that we've seen another reason for having that, which is that sometimes as problems become more challenging, we want to test your efficiency with writing the functions. So we want you to write a function that's efficient as it possibly could be. And in this particular case, speeding up pattern matching with the failure array is only going to be useful if we have a fast way of producing the failure array. Unfortunately, our approach is slow. I'm going to have to Control C and stop this process because it's not going to finish in 5 minutes. So then there's questions here. How is it that I make this faster? The approach that we have of trying all the suffixes and matching them against all the prefixes, and the first one I find, setting the failure value and breaking, that seems pretty good. This is very intuitive, and it's based off of everything that we've learned about up until this point in the series. The problem is it's not going to be fast enough to be useful in the actual KMP algorithm. So if we don't have this more efficient solution, it won't be fair to apply pattern matching, which is what we would like to do, to see how much faster is this algorithm really than the brute force approach of sliding a window and asking if the current window matches. These are questions for another time, because we could have an entire session probably just on the side of, for one, how do I pass the autograder in time? How do I write this failure array function in a way that is substantially faster? And then how is it that once I produce this, how is it that it makes it easier for me to match a pattern against a longer text in the first place? How is it that I adapt my pattern matching algorithm, something that we really haven't discussed at all? So why is this failure array useful in the first place? And how much faster does that make our algorithm compared to something that would be something that we wrote in one of the initial questions on this platform, which was SUBS? So we could have an entire session just on that and just on trying to fix up this problem. For that reason, we're just going to have to come back to it another time. So we're working on some much more advanced questions. We're starting to see stuff that's building on everything else. But for now, I'm going to have to see you next time. So I look forward to it. I can't wait. I hope you'll join me next time. And until then.